Hello everybody. On behalf of partners working on Bonyam, I'm presenting the main highlights and project finding on Bonyam during period 3. This presentation is prepared with the contribution of author from FSA, CIRAD, Buen University, ITA, NACRI, UNIHA France, and UNIHA Guadeloupe. 2 papers related to Bonyam were published in the special issue of International Journal of Food Science and Technology. The major highlight concern the trait and quality characteristics, morphologic characteristics such as size, shape, skin of tube, as well as sensorial or physical chemical characteristics were reported in these papers. As far as trait and quality characteristics were concerning, they are checked along the process of boiling yam. Concerning the raw yam, the traits such as mucilage content, water content, fresh color, skin appearance were reported in this paper. And concerning processing, the traits like discoloration status, peeling behavior, and aspect of cooking water, breaking ability with a fork during cooking were reported. And concerning the hand product to boiling yam, we report the fresh color, breaking ability, creaminess, taste, and concerning selected traits and quality characteristics, they are grouped in 2 classes as high and bad boiling yam traits. Concerning priority trait and quality characteristics for high or good boiling yam, we report the low water content, white cream and yellowish flesh, no discoloration, easy to break, or slightly sweet. Concerning the trait and quality characteristics for low boiling yam, we reported mucilage content, dark color, hard in mouth, bitter taste, or bad taste. The relative importance of selected characteristics were presented in this slide. We observe that no discoloration is the first, and followed by easy to break, fresh color, sweet taste, and low water content. In the other hand, high mucilage content is the first for bad characteristic, followed by bitter taste and harder in mouth, and at least dark color. We observe that the quality characteristics were dependent not only on variety but also on the cutting section, such as proximal, central, and distal portion. The quality characteristics we observe depend on these 2 parameters of these 2 factors of tubers, cutting section and variety. For each of these traits or quality characteristics, we develop proof of concept to understand the cooking ability. Concerning conventional cook method, we use oven. For example, for water content, we use oven drying. For color, we can use colorimeter. For discoloration, can determine by total phenol or PPU. Except this conversion method, we develop new method, rapid method, which will be presented after assuring the proof of concept. Concerning quality characteristic water content, we develop proof of concept using this prediction value of dry matters. And we have a good prediction model with coefficient of determination of 0.94. For discoloration, we develop two proof of concept using visual observation. The first is a direct observation of color change of yam when yam is peeled. The second method uses not only the visual observation, direct observation, but the visual observation is compared with the custom color targets. That's the two methods that is used for discoloration using visual observation. We also, for discoloration, develop a proof of concept using NIST prediction value of polyphenol. The performance of prediction model is also very good. We have a coefficient of determination of 0.91. We develop two proof of concept for cooking time. The first use fork to evaluate the cooking time, and we observe using fork that cooking time depends on yam variety, and it depends also on the location where yam were collected. We developed the second soap based on hyperspectral imaging, and this figure shows hyperspectral imaging of yam cooking degree in relation of water absorption during cooking. The image obtained before cooking and 10, 20, 25, 20, 30, and 35 minutes after cooking were shown in this slide. The hard red color corresponds to the maximum cooking or maximum water absorption, and the hard blue color here represents no cooking or minimum water absorption. From 10 to 35 minutes of cooking, we see the evolution of water absorption from the center to Concerning proof of concept to understanding the role of peptide and cell wall component impact on textural property, we have preliminary results. The study on cell wall polysaccharide impacts on texture property show that a large range of sugar, and this is a promising success due to the component which vary according to the variety and the range of components we have. These preliminary results will be improved to check the role of those components on the texture property of boiled yam. We also observe some correlation between sensory attributes and instrumental texture parameters. We observe that hard to break and hardness using penetrometer test is correlated. We observe that dry matter is correlated with ease to chew and so on. So some preliminary correlation can be observed between sensory parameters and instrumental texture parameters. Concerning the markers regression, we observe that the novel methods to bring the new variety which meets farmers and consumers' expectation was applied. The preliminary result showed that loci associated to some quality traits was identified. We see here starch content, protein, sugar, dry matter, and hardness were addressed already. And the starch content was validated. From this study, 6 promising varieties were obtaining, and they are in the stage of sanitation. We will see it in the future. Some elite clone from breeding program were identified. At IT, 16 clonings selected from RTB food product out of the 100 screening clonings. In Nigeria, NACRI has selected 14 promising cloning from Guinea yam variety which were planted for evaluation in 4 locations. At CIRAD level, 6 promising great yam variety are in the process of sanitation ready for. As far as participatory assessment of elite clone are concerning, 10 yam cronies were evaluated in 8 on-farm trial in 8 states in Nigeria for boiled yam, 5 from Dioscora rotundata and 5 from Dioscora lata. From the 10 yam cloning, 3 yam variety performed very good, very well on agronomic and food quality, and they were officially released in Nigeria. They consist of 2 rotundata variety and 1 alata variety. Concerning perspective for period 1, which concern change in breeding pipeline for the integration of new phenotyping method to select for quality traits, research genomic region and marker association associated to different traits were used, and they used at Sirad Gate two complementary approaches. The first, QTL analyze on two Dioscora and latter biparental population were related to dry matter, sugar, protein, starch content, and hardness. The GWAS method was also used on a diversity of panel of 15C, 56 Dioscora latter variety. At IT Gate, quantitative trait locus analysis related to dry matter, oxidative browning, starch content, RVA, and amylose content were used also. We have to work on the cell wall component and starch characterization for identifying biophysical proof of concept for predicting texture. We have also to improve the use as a proxy to predict texture and meaningless from characterization of WP2 data. We have to develop proof of concept for prediction of quality trait related to yam tuber shape using image analyze. That is the activity, summary of the activity we did on boring yam for the period 3. Thank you for your attention. Thank to all contributors. Thank you.